This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 539, recorded on March 15th. That's the Ides of March.
It is the Ides.
2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. And today is also Match Day.
Yes.
Let's explain that. Joining me today here in New York City, Dickson, Despommier.
Hello there, Vincent.
Everything well?
Perfect.
No.
Yeah, no, at this very moment? At this very moment? Yes.
Because you're sitting here chatting.
And then it will deteriorate rapidly after we finish talking.
So joining us from Madison, New Jersey, Brianne Barker.
Hi. It's nice to be back with you guys.
Hello,
excited
Brianne.
to talk about this.
Were you
Hi.
gone?
No, I was on spring break last week.
Oh, that's right. Spring break, yeah.
So I now feel like I'm, back.
We were in Iowa.
Yes.
Huh.
And I was in New York City, strangely enough.
I know. Well, you're probably better off. No, just not saying that. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. I enjoyed being in Iowa.
Yeah, you do.
And here in Ann Arbor, it's 40 degrees Fahrenheit. That translates to about 4 degrees Celsius.
It's amazing here in New York. It's 21 Celsius.
It's hot.
It's hot and it's cloudy.
Do you have any snow on the ground still, Kathy?
A little bit north facing.
We have bits.
Yeah.
Well, also joining us from western Massachusetts, Alan Dove.
Good to be here. I'm very excited. It's 55 Fahrenheit right now, 13 Celsius, but the forecast high today is 61.
Exactly.
Which will be 16.
Do you think it'll get cold again?
Of course.
Supposed to cool off again a little bit. We still have lots of snow on the ground.
Right.
Some bare patches showing. We're just about to start mud season here, so that's always an entertaining time.
Fun time.
So, Kathy, Iowa's in the Midwest, right?
Correct.
Yes.
Even I knew that.
Such a New Yorker.
I just wanted, because Kathy is clearly a Midwesterner.
You were just there, and you don't even know.
I was in the Midwest, right?
She likes it, and I think that's because it's where she was raised and currently resides. I looked at a map because I didn't realize it was quite west of Illinois.
It's a beautiful state. Mm-hmm. It's gorgeous.
And although I grew up in the Midwest, it was in Ohio, the other one that begins with vowels.
You grew up in Ohio, yeah.
I think Iowa is the Courier and Ives state for the entire world.
Iowa?
Yeah.
So the taxi driver who drove me from the airport, I asked him, is it flat? He said, oh, no. It's not like
Kansas.
What did he say, Montana or Minnesota? He said, there, if you lose your dog, you can still see it 3 days later.
That's good.
That's funny. It. So this guy said he drove Trump to the airport after one of his campaign speeches before he was a candidate. And he didn't make any comments.
Where did you fly into?
Cedar Rapids.
Cedar Rapids.
Okay, so I learned, which I didn't know, that Iowa City used to be the capital.
Right.
Mm-hmm.
And then they had a political shootout. Yeah, shootout. So what's the guy's name who came to take over the Capitol, and this woman with a cannon fought him off, and they have a statue of her, right, Kathy?
Oh, right. I don't know that I got the name.
I was joking, but they did have a shootout. Okay.
They did.
Sam Houston. He wanted to move.
Wait,
that right?
Would be
That's Texas. That is Texas.
Oh, that's what Condit started talking about, Sam Houston. There was somebody in Iowa who wanted to steal the Capitol records, all the records, and bring them to another city. And eventually they moved the Capitol to Iowa City, right?
Yes.
But this lady with a cannon, she shot off these guys and chased them away, and now they have a statue of her there, commemorate her. I forgot her name.
Yeah.
But Wendy told us that story.
I've driven through Iowa many times.
My cousin rode Ragbray, which is the bike ride across Iowa. And he said, you don't know how flat Iowa is not until you're on a bike.
This is all true. I mean, really,
That's true. That's true of every place on a bike. You really appreciate the,
What state did he tell me? Who said it's so flat if your dog runs away?
North Dakota.
Maybe Dakota.
Kansas is that way.
No.
I think it was North
Sorry,
Dakota.
my son lives in Wichita. It doesn't. It's not like that.
That's where Fargo is, right?
No, North Dakota is Fargo.
Fargo. Because he said he used to live in Fargo.
That's flat. It's so flat that they have, these pull-offs for the routes that says scenic overlook. In North Dakota, it just says scenic look.
So we're pulling out of the airport. This guy's very friendly. He goes, "So are you here for this podcast?"
Did he actually say that?
Yes, but that's only because he had just picked up Condit the hour before.
Oh no, still he
and
remembered.
I'm thinking, wow, news travels fast.
Big in Iowa.
And then he said, "Yeah, I just picked up that fella Rich an hour ago," and he told
guy
me
with the
about
deep
it.
voice.
And he said he was such a nice fella.
He is.
Yeah,
is he a bass? Does he sing bass in Barbershop Quartet? Because he sounds like a bass on the air.
You're asking the wrong person.
I guess.
Pretty sure.
Okay.
So today's match day, right?
Yes.
Which means
Very
that
exciting.
graduating medical students match with their residencies.
Yes, all of them, the entire country.
All in synchrony.
Exactly.
And why is that?
It's like fireflies all turning on at the same time, right?
Because they actually have a rational labor market in medicine, unlike science.
They do. This is all
So
true.
in medicine, the medical students, as they're about to finish up, they apply to all the residencies they want to do, and then the residencies that interviewed them will rank the candidates. Right. This was our first choice, second choice, and then the medical students rank their residencies.
Correct.
And then a computer program matches to optimize the entire system and get every residency program its top picks out of the ones that ranked it, and so forth.
Yep.
Does every medical student get a residency?
Not necessarily.
No.
And what do they do when they don't?
That's a great question.
They might hang out at their same university for a year of, I forget what it's called, but I know somebody that's doing that this year because they didn't match last year.
Well, they do infomercials on late night television.
I see.
Yeah, there's that too.
So you think eventually they all get in, some may not? After
they
all
go
that,
through the match the next year.
I know some that don't even apply.
Oh, after all that.
One of them didn't want a residency. He just wanted to start practicing medicine.
It's worth noting.
Sure.
Well, you can't actually practice medicine in most areas without at least an internship.
No, this guy was really interested
I wonder
in rural
if you
medicine.
could practice in Iowa without
rural medicine. Yes, they're desperate.
You might be able to go someplace like rural parts of Alaska.
By the way,
or
he was a very bright student, and he ended up at the Johns Hopkins School of Public Health and got an MPH. So now he does.
Cool.
He does.
So every place has a different speciality that they're really good at for residencies, right?
Yeah.
Like if you want internal medicine, you go to some places. If you want, can you do a surgical residency?
Of course.
Now, what happened?
My wife did 3 years of surgery residency.
What happened to internships? That's no more.
That's your first year of residency is your internship.
Call the intern.
I see.
Right, first year intern.
So my dad came from Italy.
Right.
He had gone to medical school and he was a physician there, and he came here to just do, I don't know what he was going to do and go back, but he never went back. So he did 1 year internship at some hospital in New Jersey.
Right.
And then he did a 4-year residency at Lincoln Hospital in the Bronx.
Right.
And then he did another 4 years of surgical residency somewhere else.
Wow. That's a long
And after 10 years, he started his practice.
Goodness.
Unfortunately, he didn't get to use it for very long.
Well, we're glad he didn't go back to Italy. Otherwise, you wouldn't be here.
That's right.
Yeah, probably not. I'd be somewhere else, right?
You would be somewhere else. That's right. Your genome would be wandering the the Italian landscape.
So Susana Lopez, who has created with her colleagues in Mexico, Paul Has Measles, has now released a German version.
Nice.
So that's in addition to English, Spanish, and French. There's also a Spanish video. You can go to YouTube and watch someone narrate it in Spanish.
I tell you, measles is really rearing its ugly
And
head.
this is annoying. I got to tell you this. Of course.
Only one thing that's annoying?
I tweeted it out last night. And a guy in Sweden says, I'm going to think about whether I should share this or not, because we don't have any measles here in Sweden.
Really?
Come on, man.
Really?
I mean,
the air doesn't blow in Sweden?
Well, he says everybody gets vaccinated, but it doesn't matter.
It doesn't matter.
Because Twitter is international, right?
Yep.
There you go.
Well, and somebody could come to Sweden with measles.
But of course they could. Yeah. All the time.
Measles is in Michigan as of this week. Anyway, it was on the news this morning.
How long does the virus last outside of a human host?
Here we go. Contemplating whether to share. I had measles as a kid, but no one has measles in Sweden nowadays since almost everyone is vaccinated. Wow, that's not nice. That's a good reason not to look ever at
Nobody
Twitter.
immigrates into Sweden anymore.
People annoy you.
Yeah.
So
Dickson, they say at least 2 hours outside of the body.
2, that's all? Well, okay.
It is ridiculously contagious during those 2 hours, though.
Exactly.
It's a productive 2 hours.
Very productive.
So we'll put a link to that,
Right.
but you can download that for free. There are many other languages coming, so stay tuned for that. Some of them require graphics, not just typing. So
Did they change the name Paul to the Spanish equivalent?
Oh,
Pablo.
Is it Pablo?
Pablo.
By the way, so now at my blog, virology.ws, I have put a permanent link to Paul Has Measles on the right-hand side, so you can always get there from there.
Good.
Great.
And I'm going to figure out how to put it up even higher on the page. So it's Paul Has Measles, Paul à la rougeole, that's French, Paul hat Masern. It's German, but I know I'm massacred. I'm
Pablo
sorry.
tiene sarampión is in Spanish.
Yes,
There you
Pablo.
go.
I like Pablo. That's just such a great name.
Especially when it's preceded by Picasso.
Yeah.
One of my pictures at home, I've got that. No, I don't, actually.
Dixon, you want to take that first?
Yeah, sure, of course. Follow up. Steen writes, and thank you, Steen, for all this. Regarding Dickson's pick of the week on TWiV 537, starting at 129.02, it looks like leafhoppers, Psychedelidae, are closely related but not distinct from tree hoppers.
No, but distinct.
Are closely related to, but distinct, right? But
That's
distinct.
not what you said.
As I said, not from,
Yeah.
They're distinct from.
They are distinct from tree hoppers, which are Membracidae. As briefly noted in the National Geographic piece, tree hoppers transmit fewer plant viruses. 2 are known: tomato pseudo-curly top virus. Right. You look at it, it's curly. No, it's not. And then grapevine red blotch virus. Both are single-stranded DNA Gemini viruses in the family which you have featured on TWiV 512 with Anne Simon. Leafhoppers also transmit several economically important Gemini viruses. See the ICT family profile, as well as other viruses such as the Rio virus featured in the paper and cover image that Dickson described. Thanks to Dickson for excellent back-to-back picks. Pedantically yours, Steen. P.S. To be a bit more explicit, no need to read this on the air. Okay, well. I'll read it. Okay.
He's just explaining a little more.
The Nat Geo piece was on tree hoppers, but Dickson said leaf hoppers at the start, and I was wrong. I clearly admit I was wrong. Everything else he said was valid, but the connection between Nat Geo piece and the PLOS Pathogens article is not quite as direct as he made it out to sound.
So you said leaf, but it should have been tree.
Yeah, I was laboring under the impression that there was no difference because I said, I've never seen a leafhopper look like that before. Now I know why.
It's a tree hopper.
Exactly. So I learned by my mistakes. I hope that I will never do that one again.
Oh, you will, but that's okay. We have people to correct us, right?
I love it. I know. In fact, you're going to have me read the next one also?
Yeah, the next letter tears you a new one.
Sure.
To err is human. To err twice is dumb.
No, but this is a different subject.
It is. It's totally different. I can err in many different subjects, by the way.
Making all new mistakes.
Yeah, that's right. Patrick writes, hello, Twivers. I just want to drop a note in to push back against Dickson's comments about homeschooled students in your recent episode. He mentioned that homeschooling undermines the country's intellectual capacity, keeps students from getting involved in debates, and isolates people. I take exception to his comments because I was homeschooled from the 4th grade through high school. I was not isolated. I participated in numerous activities, including homeschooled speech and debate league, where I engaged in debates and considered other ideas. I was not intellectually stunted. I got scholarships for debate and academics to attend a liberal arts school, Concordia University, which is an excellent place. And from there, I have gone to Baylor University, another excellent place where I am working on a molecular biology PhD. Through this process, I have met many successful homeschool students in a variety of fields. Many of the highest-ranked collegiate debaters in the country are homeschooled, including several who were on Concordia's team. Moving from anecdote to data, this USA Today article gives a nice study of a Texas A&M study on outcomes of homeschooled students in college, and then he gives the reference. It seems we do quite well. And in fact, I will interject here in passing that my very good friend Bill Campbell, who went on to win the Nobel Prize, was also homeschooled. So I have nothing against it per se. I understand that there are people who use homeschooling as a method to silo students in their intellectual conformity. That's what I object to. And it is particularly pernicious when people homeschool their children in order not to vaccinate them. I really object to that. I just wanted to point out that this does not describe most homeschooled students, and the comments implying that homeschooling imparts a lack of exposure to other ideas or intellectual rigor are inaccurate, and I'm so happy that you're straightening this out. I trust that Dickson does not make these comments out of any ill will, that is true, but perhaps just sees the bad side of this practice when it's used as an excuse for anti-vaxxers. Absolutely. I have great respect for you and all, and I love your show. I really wish I were writing to tell you about how my research deals with one of your episodes instead of this. I hope you'll excuse the long email and believe me when I say it is not in any angry tone. Certainly not as angry as all those other people who complain about the weather updates. I just wanted to inform you that there are many good and accomplished homeschooled students out there who are vaccinated and enjoy a rigorous intellectual debate. Oh, and here in Waco, it is minus 3°C, 27 Fahrenheit, and will swing all the way back up to 24°C, 75 F by Friday. Texas weather is weird.
It is.
Yeah, that was great. That was terrific, by the way.
I figured you'd get a little response.
I loved it.
But you don't have anything against homeschoolers. It's only when used as an excuse for not vaccinating your kids, right?
Like we have a super ultra-Orthodox Jewish community in New York City who's absolutely giving us fits with regards to the outbreaks of measles. Almost all the measles outbreak occurs in that community, and a lot of them are homeschooled.
So currently there's an outbreak up in Rockland County, New York State.
But it's not, the homeschooling is not the problem.
No.
That's being used to
That's exactly correct.
Yeah.
No, 100% correct.
So there's an outbreak in Rockland County, and the county will not let the kids go to school unless they're vaccinated. And the parents are suing to be able to send their kids. Oh, they lost? Because the county said, no way you're going to win this. It's lost good.
I mean, forget public health.
The thing is that when I first started in polio, I used to read all the old papers about developing vaccines. People were so ardent about protecting and helping people, and people wanted this vaccine. Right, there was so much enthusiasm.
Exactly.
And lots of people devoted their careers to it. And these individuals just throw all that out the window. Like, no, we want our...
There are certain people whose children actually died as the result of receiving the vaccine. And it's like 1 in 1 million or 1 in 2 million or 1 in 5 million. And when you start looking at the odds, what is the risk benefits? But for that one person, that was a big tragedy in their lives. And
did you see the story about the 6-year-old who got tetanus? Mm-hmm. And he had this expensive treatment and it was horrible, lasted a long time. And then as part of it, they gave him a tetanus shot right off the bat.
Texas shot.
And
big shot.
But then after all was said and done, his parents did not allow him to get boosters or any other vaccinations. Yeah. It seemed to me, I don't know, Brianne, if you got this impression that he almost died.
I had that impression. It was very serious, is what I had heard.
He now has natural immunity, right?
Oh, well, not.
No, but you should, and this is an argument many people make, we should get natural immunity. No.
Not for this one.
No.
That's how you shorten your lifespan.
It's the whole point of vaccines, to have immunity without the pathogenic consequences, right?
Right. I think it was something like $800,000 in medical costs.
Yes.
Yeah.
Wow. Yeah, yeah.
Steve writes, you were talking about backing up podcasts, and then I see this, a link to a Gizmodo article called Delete Never, the digital hoarders who collect tumblers, medieval manuscripts, and terabytes of text files. And these are people who buy hard drives and save stuff. Wow. And there's a Reddit forum called /r/data hoarder, where they trade tips on building home data servers, share collections of rare files,
Wow.
From video game manuals to ambient audio records, and discuss the best cloud services for backing up files. Well, somewhere out there, maybe someone is hoarding TWiV.
Let's hope.
So actually, I have all the old TWiV episodes. Okay?
You do? Oh.
I put them on a hard drive, and then I back up that hard drive to a second hard drive, and they're all sitting, of course, in my office, which if the house burns down, they're all gone, right?
This is true.
So it should be in the third place, but I started
Safety
uploading
deposit
some of them
box.
to, I have an account on the Amazon Cloud S3, and I started uploading it, and then the monthly cost just got too big, and I stopped doing that.
But you could at least have one of those hard drives at work. I mean, you could have it in a remote place.
So the thing is now, for TWiV, I'm on my 8th hard drive. You know, these are 1 or 2 terabyte hard drives.
Yeah.
So I could bring the earlier ones, which I don't need to access anymore here. Yes.
Right.
But Dickson, you tell me I'm leaving soon, so I might as well not bring them here.
Are you?
No, I'm just kidding.
Please don't leave.
I'm just kidding. We actually have 2 papers today, which are unusual in that they report what seem like remarkable findings that could be paradigm shifting. You know,
We're going to remark on them.
with additional research, they might be, but they're both quite unusual, I think.
They are.
And the first one is an accepted manuscript in the Journal of Virology, which is called "Chloroviruses Lure Hosts Through Long-Distance Chemical Signaling." Now, Dickson, they threw in a phishing thing there.
Yeah.
So these individuals are from the University of Nebraska and the University of Baghdad and the Middle Technical University in Baghdad. So I guess there's no phishing in Baghdad, right?
No,
So they,
Probably is.
it's on a
think so?
river. It's on a river.
Yeah.
First author is David Dunnigan. And then we have Al Samak, Al Amili, Agarkova, DeLong, and Jim Van Etten is the last author. And we've talked about chloroviruses. So this is about this idea that, you know, we think viruses bounce around randomly and hit cells, right? And that's why there's so many of them, so they can encounter a cell and then attach and get in. But this paper is suggesting that there's some luring going on. There's some chemotaxis of the host.
That's bizarre.
Yes.
Why is it bizarre? Bizarre. Yes.
Well,
It is
because yeah, we're gonna get into this.
So these are chloroviruses, right? They're large double-stranded DNA icosahedral viruses that infect algae that are within a protist, like a paramecium.
Oh, like a symbiotic.
And they have an internal lipid membrane.
a zooplankton.
Yeah, they have an internal lipid membrane. That's right.
Zooplankton.
Yeah, so it's like sort of a turducken thing.
A
Duncan.
They're
So
the-
it's an
they're the
inside-out
inside
virus. They've got a capsid and then a membrane inside it.
And we talked
It's more
about
like
them
sushi.
before because, you know, how to infect this algae when it's inside the paramecium is interesting. It turns out the whole assembly is eaten by a some kind of
They wait till the paramecium is poop.
what do you call? Yeah, they poop it out. Copepod. Thank you.
A poop-
So
Copepod?
copepod eats the paramecium, poops it out, and then the virus infects the chloroplast.
It's released, the virus and the algae is released, and then they can infect. Oh man, what a weird. Well, that's how it goes in life, right? Or
Yeah.
death, or non-life, whatever. But anyway, so now.
Sorry, not chloroplast, Zochlorelli.
So Claret, I don't know why they did this, but they did an experiment where they make these agar plates with a mold in it. So you basically have like a center well and then arms, depressions in the agar going out to peripheral wells, right?
Looks like a Mercedes logo.
Well, but if you've ever worked in a lab, they're these three-way connectors. They're the white plastic three-way.
Yeah,
Oh.
they use that to mold it, right?
Okay.
So then in the middle, you can put your paramecium,
Right.
and then in the outer wells, you put something that you think it might be attracted to. And then you watch them move, and you can see
I
these.
see what.
You give it the Pepsi challenge. Yeah.
Got it.
Yes.
Do you want to go over here? Do you want to go over here?
Yeah, it's how you test for pheromones.
Right.
Yep.
That's what they're doing.
Yes.
And so they put various things in these wells, including nothing, just buffer, a cell extract of
Right.
the Zoochlorellae,
Right.
purified virus.
Oh.
And guess what? The paramecium move to either virus-infected cells or purified virus.
Wow.
And the more virus you put, the more they move towards. Not everybody moves, but they don't move towards a control well.
Wait a minute.
No
I have to
virus
raise my
in
hand
it.
at this.
They don't move to a well with just cells without virus in it.
Right.
These are not viruses that are metabolically active, though, because there's no cell.
They're just particles.
So
Particles.
What's the signal?
So they
That's
do an
a good
experiment where they centrifuge the virus. They make a pellet and they take the supernatant.
That's a good idea,
They
Ali.
put the supernatant in one of these wells and it goes to that.
Yeah, okay, fine.
It was something shed by the virus particles.
There's some factor with the virus particles.
Right? Could be
All right.
a protein. It could. They say there are lots of small molecules inside.
They call them leaky.
Leaky.
Leaky. Yes.
Richard Leaky.
Yeah, and it made me think about the whole concept of metastable. Are they less than metastable if they're leaky?
Yeah, coming apart on their own. Yeah, who knows? So,
And these, by the way, these an observation that was known before this work and might have been the inspiration for it was that the virus is found. It piles up on the cilia of the paramecia.
right, that's right.
So that's how it.
Yeah.
It doesn't just drift across a pile of poop that used to be a paramecium that got eaten by a copepod. It's actually on the paramecium. It's like a skin microbe almost, but it's just the virus sitting there on the cilia, and there are hundreds of particles there.
And
then
then that whole thing gets eaten, right?
So
right, and the whole thing gets eaten, and then the virus is able to finally infect the algae that used to be inside the paramecia. So the question was, how did the paramecia pile up these virions? Is it just because they collided with the virus, or is something else going on? And that's
what led to this. Yeah, makes sense.
Well, it still has to attach, though, after it encounters the paramecium, right? So there must be a receptor there.
Sure, there's attachment going on. But the paramecium is swimming toward the virus.
Yeah.
Which is going to make it a whole lot easier.
Which is weird because the paramecium is not going to be actually infected. It's going to be the algae within the paramecium, yet it has evolved to do this.
And these are only paramecia with Zooxanthellae associates.
Well, they actually just use plain old paramecia, I think, right? Do they have the algae in them?
I think they use plain old paramecia in some of the experiments. I don't think they're all infected with the algae.
Because that would be interesting if the algae conferred this property, right?
It would.
Right. I mean, I think the thing that made this interesting to me was I started thinking about how spread out the paramecia and the viruses would be in a body of water in a way that there would have to be some way to concentrate them. They'd be too dilute if not for a mechanism like this.
Yeah.
Raises all
they have
kinds
a
of
mechanism
interesting
like this.
Right, exactly.
Maybe this is it. Yeah, that's a good point. So normally
might help
it would
get around that problem.
That's a good point. Yeah, that could do that.
So they're not ingested by the paramecia; they're just attached to them.
They're stuck.
So it's like a decorated paramecium.
So it seemed, as I was reading this, I was thinking, wait a second, why would the paramecium move toward this virus that doesn't seem to serve it any benefit? But I assume the virus has hit on some molecular signal that the paramecium normally is attracted to, and is hijacking that pathway.
Food.
And the next thing, right? It's for some food signal or whatever. And the next thing to do, obviously, is to isolate what these factors are that are leaking out of the virus and that the paramecium is gravitating
Well,
to.
another thing to do, if I were doing this, of course, is to see how long the signal could be produced by the virus, the same virus. So I would take the virus and put it in the well, and then, of course, they come to it.
Wash it repeatedly.
Yeah, exactly right. How long-lived is this ability to attract paramecium?
Lots of things you could do.
You could.
Yeah, I'd like to know what's coming from the virus or what's detecting it on the paramecium.
So you could put various sized filters in between.
How widespread this is.
Is there, I don't know what the techniques are available with paramecia. Is there a lot of ability to genetically engineer them?
I don't know.
Is this multi-nucleatum or just one nucleus?
Oh, Peeper Saria. I don't know.
Because there are some paramecia that have hundreds of nuclei inside.
That could complicate things.
The macro plus the micro nucleus.
They were originally collected from
I was just thinking, try and find
in Denton, Nebraska.
Oh, that I know that one. I know the paramecia.
So these are probably not a laboratory strain.
These are local paramecia.
Just something they pulled out of a creek.
Right.
And they tried different chlorella strains, and they all do the same thing. They all attract the paramecia.
Right.
Yeah.
Wow.
So this sort of, they like to think that this is a general new thing, that there can be attraction of hosts to the virus.
But you said the zoochlorella attract, but they have to be infected with the virus in order for that to work, no?
No. Paramecium.
The paramecium is gravitating to the virus.
You put purified virus and it gravitates to it. The infected cells will also attract, but I assume something's coming out of the cell.
Right.
To do
but
that.
plain algae will not attract
the
paramecium.
Right.
So this could be broader, but of course the requirement is you need a mobile host that could move, right?
Yes.
Mm-hmm.
And they mentioned that maybe macrophages or dendritic cells could also be attracted in organism, mammals, even animals. So maybe.
Do we know how the virus gets into the zoochlorella?
Yeah, it's eaten by a copepod.
And then what happens?
Well, then it's
Copepod poops it out.
It's pooped out.
I want to know what happens next.
Well, the poop in the poop you have that
nice.
that breaches the barrier of the paramecium, and so now you have the zoochlorella just sitting there in the poop, available to be infected.
And the virus binds to the zoochlorella.
Virus binds, then it can get to the zoochlorella, because the problem it's overcoming here is how to get into the paramecium.
No, I got it. So they have it isolated though. They've got pure zoochlorella with no virus. They've got pure virus.
Yep.
Can you just mix those two things together and get infected with zoochlorella?
Oh, yeah.
In fact, they can do plaque assays on monolayers of the algae. It makes green monolayers with plaques. It's beautiful.
Did we?
You don't even have to stain them.
Did we ask whether or not the infected zoochlorella can attract?
Yes, they did that. Yes.
Okay.
Yes. And extracts of infected zoochlorella work. Yes. And virus works. Yes.
Now, in terms of generalizing this, this is something that, as Brianne just commented, I think it was Brianne who just commented, that this makes perfect sense for a situation where you have this extreme dilution.
Yes,
right.
This is
whereas the idea of macrophages, being chemoattracted to virus inside a body, there's a lot less need for this type of thing. Not saying it wouldn't happen, but
macrophages are attracted to virus-infected cells.
Macrophages are attracted to foreign bodies,
right? Because the cytokines that are produced by the infected cells will attract macrophages and dendritic cells.
Paramecia are virus sponges.
Sponges.
They just soak them up from the environment.
They're decorated.
And they concentrate.
Yeah.
And then they're eaten by copepods.
So what other things?
They're sort of virus flypaper.
Yes.
What other?
I'll change that.
And a copepod comes along and eats it.
That's
so
right.
what other cells in nature, Dickson, could do this? So paramecia are clearly modal. What else? Like rotifers?
No, I think about
Plankton.
Plankton.
I want to know about other ciliates. There's a ton of other ciliates out there. I mean, euplodes. There's stationary ones called stentor. I don't know if you know.
They're all the amoebae.
Yeah, and then there are amoebae also, of course, but I don't know if amoebae have zoochlorella though. That's what I don't know.
Oh, right. You need
I think stentor. I think some forms
it
of
could work.
Be bigger than just zoochlorella.
Yeah, it could work for the amoebae because we all know these giant viruses that infect amoebae. It would be interesting to do this kind of assay and see if they migrate.
But does this virus actually need the zoochlorella in order to succeed?
Yeah, it replicates in the zoochlorella.
This one does. That's its host.
But others do not. Others just replicate in the amoeba, right?
And then you wonder whether those are producing chemoattractant cells.
So the last two sentences are, we must now rethink how virus particles can play an active role in directing their own fate. If one virus can do this, it is likely that others do as well. I would say it's not an active role. It's just all passive. They're just shedding things that happen to attract the paramecium, right?
Right.
But it'd be interesting to know how broad this is, for sure. So I at ASV last summer, I sat next to Dave Dunnigan at lunch the first day, and he said, "You're not going to believe what we just found." And I said, "Well, when it's published, let me know." And he actually told me about this a week ago.
Cool.
Because I wanted to talk about it because it's pretty neat.
Yeah.
And I must say it's a very straightforward paper, and there's a picture of the maze there.
And does the virus actually end up killing the zoochlorella in the course of its replication cycle?
Yeah, I think so. That's why you can do plaque assays.
Yeah.
So that's in vitro. What about inside the paramecium?
Yeah, I think it does.
Oh, it doesn't. But it doesn't get inside the paramecium.
No, or inside the that's shit. No, wait a minute. Where does the zoochlorella plus the virus live?
Outside when the paramecium is breached.
Yeah, right. But what about the infected zoochlorella? Is that not eaten by something else also?
No, that's the replication cycle completes. The virus gets out of the infected zoochlorella and then is drifting in the water, waiting to be picked up by another paramecium so that it can restart the cycle.
Picked up on the outside of
the outside of another paramecium.
No, right. So in the poop from the copepod, that's where the cycle takes place. And
get the cycle right. That's where you get the infection of the zoochlorella.
And what is the rate of how many particles per zoochlorella do you get for this virus?
Don't know offhand.
We didn't get that data.
It's in an older paper, I'm sure, but
all right.
Didn't do it. So would you call this a paratenic host, Dickson?
Paratenic.
No.
What? Which one?
I thought you would tell me.
No. Who is the host for this infected zoochlorella? There is no host.
It's inside the paramecium.
It's a commensal.
Inside the paramecium.
The zoochlorella uninfected is inside the paramecium. I get that part.
Yes.
But the infected zoochlorella
it's outside.
Only occurs in the copepod poop.
Yeah, I think so.
So there is no host for that part.
Right, so there's
it's a free living. I would say it's free living.
Well, it doesn't want to be free living. It gets digested in the poop.
It got thrown out of its house.
It has a diarrheic life cycle.
Yeah. Okay. Anyway, that's a very provocative finding.
Very interesting.
Mm-hmm.
Whether it endures or not, we'll see. If it's just a one-off or if there are others, we'll see. People will start doing this.
I would use this as proof that nature doesn't waste anything either. Even copepod poo.
Well, that was another paper.
Right.
And he made a little movie of that happening if you want to watch.
I think I would like to watch that.
But we did it on TWiV.
Everybody poops. Now I can show a picture of a copepod.
Do this.
Pooping.
Go to Google and do TWiV.
I will.
Copepod, and I'm guaranteeing you TWiV 416. Oh, and this is one of the best titles because Jolene Ramsey made it. Scattered Seeds Dormant.
Yes.
Wow.
Yeah.
I mean, she had sent that in a haiku.
Mm-hmm.
Okay.
And that was, it's way back when we used to have ads. Yes, the winners of the Haiku Limerick contest. That was fun.
Huh. Why don't we do that again?
And he had a movie of that. What?
Have another contest.
If we have books to give away.
Yeah, we gave something away.
I
don't remember what we gave away. But isn't Scattered Seeds Dormant wonderful?
Yes.
Mm-hmm. That is a good one.
Great. Okay.
This paper reminded me a little bit of when you guys talked about on TWiM as well, where the phages were listening in on the bacterial cells.
Mm.
So I was sort of thinking there the viruses were listening, and here the viruses are talking.
That's good.
What language are they speaking?
The universal language of reproduction.
The language of love.
The language of love. I love it. That's exactly right.
All right, so the second paper is published in eLife, and the title is A Multicellular Way of Life for a Multipartite Virus. And the multicellular is all throughout the news this week. Right? Everyone's loving this.
It was published on the 12th, and it was on Twitter that night. And
I think Stephanie tweeted it at all of us on Tuesday.
Yes.
Did she?
She did, yes.
Maybe I missed that. Anyway, Université de Montpellier, and Anne Sicard is the first author. And I will say a couple of authors just to cover myself here. Pirol, Gallet, Vernet, Yvon Urbino, Peter Schmidt, Gutierrez, Michalakis, and Stefan Blanc.
That's actually all the authors.
It's all of them. I decided to just
go for it.
Vincent, I love it when you speak French.
I can speak in Italian too. As Kathy says, I put an Italian accent on every language.
Do you ever watch The Addams Family?
Okay.
Did anyone ever watch The Addams Family?
Yes.
No,
Good.
I did, but I don't know what you're referring to.
Yes.
Oh, he liked it when she spoke French.
He sure did.
Yeah, but and then there was the other movie, A Fish Called Wanda, where he liked her to speak
exactly.
Russian or
she liked men who spoke
other languages.
That was Russian, that's right.
So this is about plant viruses where the genome is in different circles, in this case, of DNA, and they're all packaged separately. And in this case, they work on one that has 8 set genome.
8 packages.
and they're 8 separate particles.
So then the question is, how do you get infection?
That's bizarre.
You have to get
and
all
is
8
bizarre.
segments into one cell.
Right.
Perhaps.
So when I first started reading this, I thought, oh, right, this is one of these weird plant virus obscure things. It's got separate encapsulated pieces. And then I saw this sentence in the intro that plants and fungi, like 35 to 40% of the viral genera and families described in plants and fungi operate this way.
That's incredible.
And I think it's
This is
only
not
in
some
plants.
corner case.
This is only in plants, though. I looked
Right.
elsewhere. They're not elsewhere. All right. And
I mean, that sentence says rarely in insects and
Rarely in
hypothetically in
insects.
vertebrates.
Right.
hypothetically invertebrates. Yeah,
Yeah.
but there are no vertebrate viruses.
You'd assume these are old, ancient viruses?
Why would you do that?
Because plants came before animals.
Maybe. But there are Gemini viruses where they're actually, the capsids are fused. So they always both,
Right.
and each capsid has a separate genome, but they're fused.
Right.
Hence Gemini, right?
So they never get lost.
Yeah,
And
well,
of
they
course,
both
you
can infect the same cell. Now, when
Lots of
students,
viruses that have separate segments of genome, like flu, but that's all in one capsid.
so when students hear about this, they say, well, how did they
Exactly.
do this? I said, well, we always say they have to infect the same cell because that's how we look at things in a one cell business.
There's an old joke that goes something like, how do two mating elephants find each other in the tall grass?
They find each other delightful.
Exactly.
I've heard that so many
That's the right answer. So maybe that's what the answer is here too. How these virus particles find each other delightful.
Well, you see, the authors say, nobody's ever considered how this would work. And I don't agree with that because, look, if you have a high multiplicity, let's say 5, you can infect every cell with all 8 if you have a high MOI of each
Right.
particle. So it's not impossible. They say, the evolutionary constraints are too much. I don't think so. And as we'll see, I think maybe their solution may also be asking for a lot.
If you ablate one of these 8 replication cycle centers and only have 7 left, what happens then?
Well, so the way they do this is they have 8 plasmids, which they can introduce into plants, right? And you get out virus particles.
Yeah.
So I would guess that if you took one away, that was an essential one, you wouldn't get virus particles.
Right.
Mm-hmm.
But that explains everything. Right?
Well, each circle, so these are single-stranded DNA circles. Each circle has one gene, and it's essential. They're all essential.
I understand the rules of the game. I just don't see the game happening.
What game?
That game. How do you get together?
Yeah, that's
all right,
exactly.
I understood that.
No, but let me say my idea here. So these particular
I don't know who's
viruses
on third.
are transmitted by aphids. They bite the stem, right, in order to get some sap. And in the process, they introduce a lot of virus particles in a small area. So that's a high MOI, right?
Yeah.
Yep.
So I could see that happening. Why not?
Sure.
Why not?
Right.
Sure.
Another answer to your question, Dickson, I thought there were a few sort of funny sentences in the intro, and one of them is, the benefits of this are unclear and debated.
Right. This is what we're doing right now.
Yeah, why would you put all your genomes in separate particles? Yes, I don't know. But sometimes it's not obvious.
Apparently,
not obvious to
they
us.
do it a lot because it's a popular way of replicating.
Just because we don't know doesn't mean that
Exactly.
it makes sense, right?
Exactly.
So their idea is maybe each segment goes into a separate cell in the organism, in this case, a plant, right?
Mm-hmm.
And in the end, what they're going to suggest is that, yes, that's the way it works. And then the products, either mRNAs or proteins of each segment, diffuse out and diffuse into all the cells, and that's how the virus replicates.
So it replicates simultaneously in multiple cells, and the cells exchange the proteins that they need to complete the virus replication.
Sure,
Exactly.
Sure. So the centers for replication must be pretty close to each other then.
Well, they
Not necessarily. Stuff can travel from root to
so
leaf.
this is
Yeah.
this is because it's transmitted by an aphid bite. This
in
is
the
going
phloem.
into the veins of the plant, the phloem.
Phloem, yeah.
And so the stuff that gets secreted from the cells along the phloem is going to flow through the whole plant, and so it could
I
be
see.
transmitted
I see.
wherever the virus is replicating the rest of itself.
Wow.
Sentence in the paper, proteins can move from cell to cell or long distance, root to shoot.
Root to shoot.
Shoot.
Yep.
Yes.
I like that. It's a good title, Root to Shoot.
Shoot.
And so now in you and I, Dickson, if I injected green fluorescent protein into your vein,
Yeah.
it would go everywhere, right?
believe it would.
I may not get through
I have no
brain,
restrictions.
but
I don't think the protein would not get through my brain.
Plants don't have a blood-brain barrier.
sometimes I have trouble getting through to your brain. But plants have less of an issue, right?
Much less of an issue. They mention in the discussion something about symplastic domains, and I asked a botanist in my department what that meant. And it basically sounds like there are regions of the plant that have holes in their cell wall that allow them to have contiguous cytoplasm.
Wow.
And so these domains are sort of the regions of the plant where the cytoplasm is continuous.
So the plant is one big giant syncytium.
So his exact words were, the plants think it's both beneficial and not beneficial to have a cell wall.
Oh, that.
Yes.
Right.
So plants are. So in other words, plants are the one host that could support this multicellularity of virus infection, right?
Exactly. So I would like to know what we call a cell instead of what we call a virus.
That brings up an interesting point, because if the cell cytoplasms are all connected and they're going to say, well, this particle went into that cell and this particle went into that cell, does that really matter?
And do you remember Robert Hooke's original description of a cell? I do.
Go ahead, tell us.
It was on a microscopic section of cork.
Yeah.
Which is, of course, a plant.
Yeah.
So he was unaware of what cells really were in plants.
Yeah, what they're looking at here in their microscopy looks pretty much like
cells, right?
Cells.
Yeah.
They do look like cells.
Yeah. So I guess this could happen. I just don't know why both couldn't happen, both scenarios.
The system is so foreign to animal culture cells that it's hard to think.
And you know what else I thought? I said, okay, plant virologist, what have you been waiting for all these years? I mean,
bring this on us.
If all the cells are porous, or many of them, why didn't you do this experiment before?
Right.
Oh,
So, well, now you
That's
can
just
actually do an interesting experiment because you can do plant grafting.
Well, actually, there are very few experiments in this paper, and there are a lot more that you could do.
So, Vincent, here's the deal. You have 10 or 8 genomes separated. You infect 8 plants.
With one.
With one.
And then
each.
you take and graft them.
Then you start grafting.
Well, by
viruses.
by their results, they would say you would get virus out by that.
And for how long after? You can do all kinds of wonderful things. I think high school students should be doing this for their science fair projects. This would be great.
So the technical aspect of what they did here, they introduced the genome pieces into the plants, and then they stained pairs of segments with either green or red.
Yeah, and I saw the pictures. I was fascinated by it.
And you get some cells light up green, some cells light up red, when you tag one gene red and one gene green. And if they did their controls. But the whole thing is pretty much done with this type of introduce the genomes, look where the genomes are, and then there's some subsequent experiments to determine that what seems to be going on is protein transfer between the cells. What's not in the paper, and what I would have liked to see, is some actual infections.
Yeah, I think this is a big weakness, because they say in the materials, all these papers have done this and they see infectious virus. Since when is that acceptable?
Well, and okay, so other papers have done this infection and seen infectious virus, but have they seen virus infection starting naturally in different cells that you then fish label like you did in your transduction experiment? Duplicate this whole experiment, but with actual virus.
So that's the thing. What they do is they initiate the infection by introducing 8 plasmids into plants. Plasmids, not virus, right? And then, as Alan said, they stain for individual segments with different colors. And what they see is there's never more than one segment in a particular cell. And they do pairwise colorings, right?
Yeah.
And they do 7 of the 28 possible pairs of segments, because they only have 2 colors. They have red and green. And they typically see 1 segment per cell, never 8 segments per cell. Well, because they're only looking for 2.
Does this mean that when the first one establishes, it excludes all the others?
That's a good question.
That could be.
That's a good question.
Or it could mean that their transfection is only getting a molecule per cell.
Right.
Because I would argue that if you did this with virus and an aphid, you're going to get a high local MOI, which is
yes.
Wait a minute. Maybe that's the key to this understanding.
But this is not infection. This is
transmission.
No, I got it. But what if the aphid ingests all of the genomes?
Yeah, I'm sure it does, right?
I'm sure
Then
it does,
when it bites another plant, it's going to inject them all together.
Yeah, exactly. It's going to inject all into one place at a high multiple.
I would like them to get virus and inject it into the stem or whatever they want to inject it to, and then do the same experiment and show that.
Right, that's right.
And or in this case, where they transfected, to actually grind up the stem and show there's infectious virus there.
Right. We don't see infectious virus coming out either, not going in.
Because they say, okay, there's cells all over that have this genome here, this genome there, never all in one cell. Okay, that's great, but do they make infectious virus? Right. We don't know the answer to that. And I'm kind of stunned that this paper is published without that.
Remind me again as to which plant they're using?
Fava bean.
Fava
bean. Because the laboratory model for plant genetics is Arabidopsis.
Well, yes.
Yes, right. They use a faba bean necrotic stunt virus.
Of course they do.
Because they had these, right.
So
they needed a tractable one of these multipartite viruses, and this is what they had.
So,
Kathy first.
So what I was going to say is that, part of this rests on their sensitivity argument. So they design an approach, they think, where the detection limit becomes irrelevant.
Mm-hmm.
Right.
But I'm not sure I was totally convinced, because I would have liked to have seen a control where they had a single cell with all 8 segments in it to show that they could detect all 8 segments simultaneously.
Right. Good idea.
Yes.
Very good idea.
And so it seems to me there are single cell approaches that might have allowed them to do that. And even though it is nucleic acid labeling, so it's really sensitive. They're not just detecting the proteins. They're doing this fish, so that's about as sensitive as you can get. I still wanted to know that they could rule out that there weren't 8 segments in a cell.
Yeah. And with that sensitivity argument, especially in the third figure, I don't know how much detail we're going to go through some of these. They kind of are saying that because they are able to detect this DNA, that DNA must have been replicated.
Yeah.
Right.
Yeah.
It's a big part of one of the figures. So they don't really talk about what if there was that low amount of DNA that wasn't replicated.
Yeah. I have a generalist question here relating to the Luther Burbank approach to creating new vegetables and fruits. And then I want to know something about viral specificity. So you can take the branch of any tree and graft it to the branch of any other tree, and you can get that to take, and you can have multiple fruits growing on the same tree. Right?
Right.
Mm-hmm.
What restricts these viruses from going into different plants?
Vectors.
Receptor tropism?
I mean, there's nothing restricting an entire organism from going into another plant.
There are no receptors, right? These are vectored mostly or introduced by damage into plants.
So my question then would be
worms, insects. And
here's the fava bean. Here's Arabidopsis.
Yeah.
Infected fava bean, not infected Arabidopsis. You graft them together. Does the Arabidopsis become infected?
Can you graft those plants?
I have no clue as to whether that's possible.
I know you can graft trees. I don't know if you can reliably graft.
There's no histocompatibility problems with these.
No, they don't have an immune problem. I'm just wondering if they have a vascularity problem. I mean, you've snipped an Arabidopsis is a tiny little plant.
Yeah.
Brianne, can you grab your botanist colleague there?
I can go get him. I can go down the hall and find him.
Ask if you can graft 2 plants.
Okay.
A bean plant to a mustard plant.
I mean,
sounds like a good dish.
Luther Burbank didn't know whether he could or not. He just did it, and it worked.
So we, as Brianne said, one of the things they conclude is that either one or a very few copy of a DNA segment initially enters cells and then replicates.
There must be an exclusion.
This again underscores the MOI, the multiplicity of infection problem.
Yeah, there's an exclusion problem.
Suggests that again, maybe the reason they're only seeing one of these genes per, these genes segregating to different cells is that they went in at such a low concentration that they're only getting a couple of molecules per cell, and you would see this segregation pattern, and it's not a natural infection.
I mean, if they just looked for virus, that would really solve. If they said we get infectious virus out of this, okay, then under these conditions, I believe it. But
you can get infectious virus, right?
they didn't, and that's a big problem.
And on the input side of it, one of the things that came up for me reading this was, okay, they're presenting this as a new paradigm, this whole new, and if you take it at face value, it's pretty freaking mind-blowing, right? That a virus, if their theory is correct, a virus multiplying simultaneously in multiple cells and exchanging material in this multicellular approach, that's radical.
Yeah, for sure.
Right?
Yeah.
That's just really, really different from the way we think about viruses working.
Yeah.
But for that to be that interesting, it would have to be the main way that this virus does its business.
Yeah. Yeah.
But it's possible that normally in the high multiplicity infection, when the aphid spits this thing into the plant,
yeah, sure,
you get all 8 pieces in a cell, and the replication happens that way. And this exchange of proteins may have evolved either just as a byproduct, it happens, or it could be a backup system. Like maybe if you only get 6 of these into a cell, it can absorb the missing 2 components that it needs from adjacent cells, which is a whole lot less interesting. It's not uninteresting, but it's less interesting.
Agree. I think it's highly possible that both modalities can occur. Individual segments in cells and plants, and all the products come out and smush around, and eight segments, or maybe even 4 and 4, according to them.
And even if it's low probability to get all 8, if you got all 8, it seems it would be more highly efficient than the smushing around method.
Yeah. That's what I think. They say this makes more evolutionary sense than the 8 getting into 1, but I don't think so. I don't see why it does, right?
And I certainly don't get why there would be an advantage of excluding others from the cell.
Right.
Yeah. So you have to wonder, would you get 1 to 2 segments per cell, right?
Right.
It seems that would be favored by very, very low inocula of DNA or something like that. Right?
Yeah.
Well, another thing to know is how long after the DNA plasmids get inside the cell do they last?
So they say that the circles may come and go, actually, because they find, some evidence where the protein product of one is present, but the DNA is
missing.
Yeah, I thought that was sort of a clever experiment. I had a lot of things I thought about that experiment, actually. So they were focusing on 2 of the segments, and one of those 2 segments includes R, is R, which looks to me to be the polymerase.
Mm-hmm.
And so they look to see whether or not the polymerase protein is in the cells that have high amounts of the other nucleic acid, making the assumption that the polymerase must have replicated that nucleic acid.
Right.
And so I think that was where they were trying to talk about the protein transfer, and they show that they can find cells that have a different type of nucleic acid. They don't have the R polymerase nucleic acid, but they do have polymerase protein.
Yeah.
And
so
I just wanted to.
They said the protein could be found in 85% of cells, but the genome was only in a minority of cells.
Exactly.
So they say that either the mRNA or the protein must be getting out of the cell and getting into other cells. Right?
What does the capsid look like?
I think it's icosahedral.
These are icosahedral capsids.
Small.
There are 8 circles inside.
No. Each capsid has
one
single circle. That's the whole point here.
That
right?
they're packaged.
No, that's that. Okay.
They're packaged separately. Yeah.
Called nanoviruses.
I did have the question though of whether or not they were sure that there were no host cell polymerases that could replicate this DNA.
Right.
And that might be something they knew beforehand that I just don't know about this virus.
Point if it's a DNA virus.
Right.
Yeah. So this, what is it? The M protein? M-rep. You think that's the polymerase? Yeah.
I inferred from what I read about Figure 3 that was the polymerase.
Actually, there is a paragraph here where they describe this.
Describe what they all are. It's in the Figure Legend 1. Oh, this is open access, by the way.
Yeah, it is.
It's in eLife.
So replication is segment R encoding M-rep. Encapsidation is segment S encoding CP capsid protein, and intra-host movement segment M encoding the movement protein.
It's even more bizarre.
And the way they talk about figure 3, it sounds like they're calling MREP a polymerase.
Mm-hmm.
I hate to say,
well, it could be it's a DNA polymerase, you think, or a RNA polymerase?
It's a DNA virus.
Probably a DNA polymerase.
It's almost like watching the results from the lottery where the ping pong balls come up. When you need 8 in order to win, and you get 7, and then the last one comes into place, bing, you got yourself an infection. But each one of those is a separate infection.
Yeah.
That's wild. There's no human or animal virus like that at all.
I looked, there are no most.
You said the Gemini viruses are linked.
It's a plant.
That's a plant virus too. Yeah.
Mm-hmm.
So the replication scheme, the viral DNA single-stranded gets in the nucleus. It's converted to double-stranded DNA with the participation of cellular proteins, which is typical. Mm-hmm. Transcription, because you need to make viral proteins, including the rep, which then will replicate the genome by rolling circle. Yeah.
Right. So rep is not a polymerase, is it?
Probably not. It says
I'm on the viral zone page.
That's exactly
this
what I'm reading now.
complicates it a little bit. So there's
me too.
para-rep and there's master rep.
Right.
Para-rep are satellite rep proteins that are only able to initiate replication of their genomic DNA. Unlike genomic encoded master rep, which promotes replication of all viral genomic single-strand DNAs, trans replication. So
under the replication description, it's initiated by cleavage of the positive strand by rep, and then rolling circle produces single-strand genomes. So it's replicating with host enzymes, I think.
Would have to.
Yeah.
Mm-hmm.
So I'm sure that Steen will write us and correct all this. But the basic issues still remain, that, and we've discussed those, that there are lots of questions here.
DNA is not virus.
It's a provocative observation.
So wait, but one more question.
I'm not leaving.
Each one of these circles has to at least encode for the capsid plus itself.
Well, one of them does for the capsid.
No, it doesn't have. No, because
where does the capsid protein come from then?
Capsid gets made by one of them and then transferred to the other
cell.
And
then it all just sort of finds each other?
Yes, that's the
smushiness.
Smushy.
Yeah. Isn't that weird? I mean,
the cells exchange the components.
Weird. But, yes, sometimes in science, weird things are okay, but...
What does David Baltimore think of all this?
Well, I don't know. He probably hasn't even read it. Most virologists don't think outside their own virus.
It doesn't violate his 7 replication cycles.
They don't think outside the capsid.
They don't think outside the capsid.
But yeah, it all has to diffuse, and every cell has to get everything, which seems as hard as getting 8 particles to infect one cell, in my view, but that's my human view,
Yeah.
Viruses don't care if it's hard.
Well, they don't.
For them,
evolution. It's
hard means nothing.
Anyway, this needs more experiments, and there's a lot of issues here. And so,
it is a very interesting paper, though.
It's potentially very interesting. But all the headlines are based on the assumption that it's true, which
yes,
we know may
not
be.
We're not quite there yet.
So it kind of, but that's the way the news cycle works, right? People get excited about something.
Sure.
Yeah, they made it sound like it represented all viruses instead of being sort of one interesting example.
Yeah. I mean, I would say if this holds true in plants, that's because plants have this strange,
crazy cellular arrangement. That's right.
And that's fine. And that's fine.
But if it holds true in plants, and given the stat at the top that this is, 35 to 40% of viral genera in plants and fungi, this would be a pretty big deal.
It would be. And
it would be a really, really new way to think about viruses replicating, if true.
And it's, as you said, even if it can go either way, 8 viruses in one cell or 8 viruses scattered among multi-cells, still the fact that you could do both would be interesting. Now, it would depend on how often each one occurs and what fraction of infection.
Yeah, if this that they're proposing is the main attraction, if this is the main way the virus
that would be cool.
replicates, that would be pretty mind-blowing. If this is a sidelight, a byproduct of other things that the virus does, then it's still interesting, but it's a much smaller finding.
Yeah, we'll see. We'll see. Anyway,
and at this point, we just don't know.
2 interesting, brand new, different observations. That doesn't happen every week here on TWiV.
No.
And we like that.
2 times I at least read the paper and thought, whoa.
Yeah.
Right. And if you don't work with the system, it's almost impossible to envision this without asking stupid questions. I'm not kidding. Because, I mean, you'd say, well, how the hell does the capsid get to the... Well, it just diffuses and gets out. Isn't there a mobilization protein associated with this also?
Yeah, there is. One of those
and
circles.
What does that do?
Moves things around.
Yeah, it moves what things around?
Movement protein.
Yeah, what?
They don't say that in the figure legend.
No, they don't. So what if that just mobilizes all the proteins in the whole plant so that the virus can fight each other?
Could be.
I mean, just speculating, but I don't know.
I still can't believe they were not required to do an infectivity assay by the reviewers. I have to work on plant viruses. Let's read some email. Kathy, can you take Mike's?
Oh,
No?
let's see. There we go. Yep. Mike writes, Dear TWiV hosts, I would like to follow up on your discussion of the vaccine perspectives for the flavivirus NS1 protein. I was actively involved in studying protective properties of the tick-borne encephalitis virus NS1 protein during my PhD thesis work at Chimukov Institute of Poliomyelitis and Viral Encephalitides in Moscow, Russia. In 1994, Dr. John Stevenson and colleagues, then from the PHLS Center for Applied Microbiology and Research in Salisbury, UK, demonstrated that replication-defective adenovirus vector, hello, Dr. Spindler, expressing the TBEV NS1 elicits protection against lethal TBEV in mice. He gives a link. Later, this project had been further developed by my mentor, Dr. Andrei Timofeyev, in collaboration with Dr. Stevenson. The results of this project demonstrated that 1. Plasmid-carrying NS1 gene protects against lethal TBEV challenge in the mouse model, tick-borne encephalitis virus. Heterologous vaccination with the vaccinia virus vector, hello, Dr. Condit, carrying NS1 gene and the plasmid carrying NS1 gene shows protection against TBEV challenge in the mouse model. 3. Peptide from NS1 protein elicits partial protection against TBEV challenge in mice that is mediated by the anti-NS1 antibodies. We did not look at any vascular pathology in immunized animals, but no side effects of immunization were ever observed. However, mice are not the most eloquent conversationalists, thus our conclusions regarding mouse health were based on superficial observations. I provided a short list of the publications related to this project. My impact was very modest at best, but since then, flavivirus have become the most amazing viruses for me. And he gives a link to 10 citations in PubMed. You also discussed the complexity of the vascular endothelium. I had the privilege to study the role of cytomegalovirus and obesity in the mouse model of endothelial dysfunction at LSU Health Sciences Center in Shreveport, Louisiana, under the guidance of Dr. Karen Stokes. Interactions between vascular endothelium, white blood cells, platelets, and the roles of immune responses in these interactions are indeed exquisitely intricate and complex. I hope that the group of Dr. Harris will be able to fully elucidate mechanisms underlying NS1-induced endothelial permeability. Lastly, thank you very much for your podcasts. They are a great source for my continuous education as the microbiology instructor. By the way, you don't have to say my full Russian name when you read this letter. I've been called Mike for all 13 plus years in the U.S. In case you're interested, I attached the audio file with the proper Russian pronunciation of my first name. He gives the link. Yours sincerely, Mike. And I listened to it about 5 times, and it sounded about like Mikhail.
Yeah, that's it.
Yes,
You got
Mikhail.
it. Perfect. Mikhail, now in Ohio.
Kirtland, Ohio at Lakeland Community College.
Cool.
And then he had a follow-up. Why don't you?
Oh, right. So he writes back, Dear TWiV host, this is a follow-up to a follow-up I sent in recently. First, I'd like to apologize for misspelling the last name of Dr. Condit. Here, I correct myself. I also would like to submit a listener's pick due to the growing agitation around the biopic dedicated to Queen, namely Bohemian Rhapsody, the song Postdoc Me Now. Hope you like it. And this is a really cool video with the words on it and all kinds of action through the lab. It is really well done.
Yes.
Some very talented people there.
No question.
So there's a lot of agitation around this Bohemian Rhapsody? I don't know.
Yes, there is.
What's wrong? People don't...
I think there are a few things that people are upset about. The one that I've heard the most about is they don't like how AIDS was not focused on as much as people think it should have been in Freddie Mercury's life. But I think there are a few things people are upset about.
Oh.
I just hear more from the AIDS folks since I do HIV work.
Okay. Alan, want to take the next one?
Sure. Andrew writes, "Hello, TWiV. I'm a new listener that found your podcast shortly after beginning a postdoc researching RNA viruses. Since my background is not virology, your podcast has helped better my understanding of all things viruses. Thank you. After listening to episode 535, I was thinking that FlaviNS1 may be a useful tool to permeate specific cells, maybe even for drug targeting. I'm curious what you think of this idea and the best available methods to target infected cells in general. Thanks again." And P.S. today, it's 18 centigrade with fairly clear skies.
Well, if I'm remembering, NS1 loosens the junctions, right? It's not
I think that's right.
It's not permeabilizing
so.
the cells, so that wouldn't be a good application to permeate specific cells. It's the junctions that are loosened up, Andrew.
Yes.
Okay.
But if you do, there are many proteins that do permeate, permeabilize cells, and you don't want to go doing that unless you can do it very specifically for drug targeting, because your cells need to keep their insides inside.
Now, Andrew, look into herpes virus VP22, which is an interesting protein that can get into cells, and people are attaching things to it to get them into cells, and there are others as well. We have an email from Anthony who sends a link to an Al Jazeera article. Vaccine hesitancy is not a Muslim problem, but it does seem to be a European one. Hostility towards vaccination programs is often portrayed as a problem that predominantly afflicts Muslim majority species.
Societies.
Sorry. I'm sorry.
We're in sociology now, not biology.
You'll edit that, I'm sure.
Well, as someone once commented on YouTube, I have no right talking about sociology because I'm not a sociologist.
But you're part of society.
You live in society.
That's right.
Vaccine hesitancy, however, is a major public health issue in Europe. So this is an article which, the Muslim world is frequently characterized as this epicenter of vaccine hesitancy. Actually, I never thought that.
I didn't think that.
Either.
No, I didn't think that either.
I mean, we know that there are issues.
I think it depends on which media you're reading, and maybe in the European media, it's portrayed that way. Like we Europeans are more sophisticated than this, but or something like that.
Yeah.
But no, I think of this as a global issue that specifically affects some areas, and it's a huge thing here in the U.S. We have a diverse population, and we've got a lot of people who just will not stick to facts on vaccines.
Bob sends in a link to an article in the Washington Post. The U.S. is funding dangerous experiments it doesn't want you to know about. Listen to your podcast every week. Regards, Bob. And the Forbes, and I think another paper. I think the Times also had. So basically, the idea here is that the government is now allowing the avian influenza gain of function experiments in ferrets to proceed after a lengthy review process, which has been completely transparent as opposed to what these articles say. And these articles say this because, what's his name? Mark Lipsitch.
Oh, yeah.
He's complaining. He's the author of the Post. Yeah. His opinion. He says, the government hasn't said anything. This is totally intransparent. Not true. It's all out there. The only thing not out there was the review of the grant proposals, which is never public. Only if they're funded do you see, who gets what. And that's how we found out that Kawaoka and Fouchier have both been funded to proceed with these experiments after a lengthy National Academy workshops, a cost-benefit analysis by an independent agency. So I just disagree entirely with the message sent by these. I wrote a letter to the Times, which of course they did not publish because they don't like to be told that they are wrong. But so that's fine. Well, I'll publish it somewhere else.
Guardian, try the Guardian. They flop back and forth on lots of issues. They're not afraid to admit they were wrong, really.
Or to just pretend they didn't say the previous thing.
No, but it's there.
All right. Brianne, you haven't read, right?
I have not.
Go ahead, Ryan.
Ryan writes, "Hey Vincent and the TWiV crew. I recommend watching the Jeff Holiday rants because he's been trying to get non-biology majors to understand the dangers of listening to Andrew Wakefield and his political supporters like Larry Cook in the midst of the measles outbreak." And Ryan gives us a few different links to YouTube videos about these. In another series of videos, Jeff Holiday gives us a warning as far back as 2018 that the measles outbreak would end up as a national issue in the USA. In that video, Jeff Holiday named Dell, Bigtree, and InfoWars for sparking the measles outbreak. But at the time of the rant, Jeff Holiday didn't have the exact victim numbers of the 2019 outbreak, except he accuses InfoWars for making the anti-vax movement a political issue as far back as 2018. I think it was a political issue even before 2018.
Yes.
Mm-hmm. Mm-hmm. I haven't looked at these. Has anyone?
I haven't.
I haven't looked.
Not.
I'm afraid of rants.
Right.
Rants on YouTube, even if they're correct, can be hazardous.
Even if they're rants I agree with, they can be problematic.
Rants are too loaded with emotion.
Yeah.
They're not Spock-like.
We never are, right?
So what we're saying is the rants are too damn high.
There you go. That's right. The raves, on the other hand.
Dickson, are you ready for
I'm ready.
Another one?
Ian writes, measles and antibiotics, not in America. I note that antibiotics are useful in treatment of measles. They do not, of course, affect the virus, but a substantial fraction of people infected with measles who would die or be blinded without access to health care would be fine with access to it. 2 vitamin A tablets reduces mortality by 50%, as well as morbidities, blindness. Oral rehydration and antibiotics for secondary infections greatly help too. And then he gives a link to an internet piece of data, I guess. It is important to stress that viruses cannot be directly targeted by antibiotics, but unless the death is directly due to the virus, rabies, for instance, saying it is untreatable or that good health care post-infection does not matter is going too far. Then he gives a link to the CDC's. In MMR rather. The CDC notes that the MMR vaccine is also useful as post-exposure prophylaxis up to 72 hours after exposure. From a Scotland experiencing perhaps its first sunset of the year, Ian, thanks again for the tremendous resource. Have another glass of scotch.
This was in reference to the thing we reported to last time where someone said nobody's going to die from measles in the U.S. We have antibiotics.
Right.
Exactly
Right,
right.
yeah.
Exactly right. Exactly right.
Right. So antibiotics could be useful in treating secondary infections and improving the prognosis, but
Correct.
they do not treat the virus. And
No.
the point that the politician was trying to make was we would treat measles with antibiotics, and that is not correct.
That's exactly right.
Correct.
All right, last one. Alan, you can take that one.
Sure. On TWiV 537, you mentioned anti-vax Facebook posts going viral. Was that meant to be a joke?
I don't know if I said
We'll
that.
say
Did
yes.
Of course it was meant to be
I would never joke about Facebook.
When in doubt, just say that it's
never joke about Facebook. That's right.
Breaking up Facebook won't accomplish anything. The problem is that people in general are stupid, and electronic media gives them infinite and instantaneous ways to leverage that. And he linked to a Wired interview from 1996 with Steve Jobs, and quoting from that, "When you're young, you look at television and think, there's a conspiracy. The networks have conspired to dumb us down. But when you get a little older, you realize that's not true. The networks are in business to give people exactly what they want."
Right.
That's a far more depressing thought.
Indeed.
True.
Yeah.
And then Anthony continues, education and communication won't help curtail the anti-vaccine movement either. What's needed is litigation, prosecution, and incarceration. I guess that since the Model T, people knew that drinking and driving were a bad mix. They still did it. Bartenders and hosts knew it was wrong to give alcohol to somebody visibly inebriated. They still did it. When people sued and juries handed out big sums, bartenders and hosts learned responsibility. When drunks were apprehended, fined, licenses revoked, and in the case of accidents, jailed, drunk driving diminished. For the edges of anti-vaxxers, marketing will work. Those thinking it trendy to let their children get sick can be convinced by advertising that it's really not fashionable. The PETA anti-fur ads are an example.
So we need pro-vaccine ads. Is that the idea here?
I think that's been tried.
I think so.
Who's tried it, Alan?
Um,
Like CDC or something?
yeah, CDC, various public health groups.
Well, I think, and we've mentioned this before, but you need a famous personality to make an ad and say, you should get this vaccine. Here's what it did for me. It helped save my child, et cetera, et cetera. Right. Someone famous who, because people love celebrities, right? So get them.
I've heard the comment, and I don't know who originally said this, but we would have no problem with anti-vaccination movements or any vaccine hesitancy if the millions of people whose lives had been saved by vaccines knew who they were.
Right.
Yeah, very true.
This would not come up because you would never be standing, in a group and not have somebody pipe up to say, I'd be dead if it wasn't for vaccines. But people don't know that their lives were saved by vaccines because that's the way they work, is that you don't get the disease. And so it becomes a really hard thing to sell.
Yeah.
Of course, now it's become a issue of I have a right to do with my body as I wish, right? It's because the science you cannot any longer dispute, really. So people want the right, to which I say you cannot, because you're impacting the lives of other people.
You do not have the right to hurt other people.
This is correct. Some of the most effective ads I've seen in the health field recently have involved people who the ad divides the same sentence into two parts. One is a healthy person saying it, and for one reason, and then you see another person laying in bed that can barely move. He says, I really have trouble getting out of bed every morning. And then the other one, who's suffering from lung cancer, says, I wish I could get out of bed even once. And then there's another one that comes on, and another one. Those are very, very heavy ads that really have a great effect. We don't have anything like that for vaccines. Just listening to a small child cough.
There is
whooping cough.
Well, there is actually, I've picked this group before, Families Fighting Flu.
Yeah.
Yes, right.
And I think it ought to be required for people who are saying, oh, gee, I don't know if I want my kid to get all their vaccines. They should have to watch some of the ads from that group, which, if you're a parent, are very, very hard to sit through without getting pretty worked up.
Yeah, that's
our diphtheria is present.
Because those are examples where people actually do know the outcome of not vaccinating.
Oh, but of course. Absolutely. And whooping cough, I can't think of anything worse than hearing a 2-year-old kid. They're not going to die from it, maybe, but boy, they sure sound like it.
Or the personal stories. I have a friend 20 years younger than I, and her child died at age 2 of a vaccine preventable disease.
Precisely.
It's devastating.
Yes, it is.
When I was 8, my family brought me to Italy for the first time, and we stayed with my father's family in a remote mountainous region in the middle of nowhere, right? And a bunch of us kids were all put in one room. And there was and my cousin, she had whooping cough, and she coughed all night.
Sure.
I'll never forget the cough.
Because
I said to my mother, what is that?
It's incredible.
I never heard it. She said, it's whooping cough. I said, well, am I going to get it? She said, no, you were vaccinated.
Right.
Because at the time they weren't vaccinating in Italy. Well, fortunately, she survived. But for me, that was like, wow.
Exactly.
I never heard any kid with that kind of cough. It's really strange. All right, let's do some picks.
Sure.
Brianne, what do you have for us?
I came across an article this week that's called "Why Do So Many Scientists Want to Be Filmmakers?" And it starts out, it's from Nautilus, and it starts out with this discussion where Nautilus had asked scientists what they would do if they weren't a scientist, just like Vincent always does in interviews. And they said that things related to movies, filmmakers, directors were sort of the most common answer. And then the rest of the article goes into talking about the relationship between science and art, which we've, of course, talked about quite a few times before, and why scientists might be captured by the idea of making art. There are a couple of great quotes that I thought Dickson would like, that both science and art are creativity and imagination and execution. And the purpose of science is to feed the soul in the same way that art does. So I thought it was a really nice discussion, summarizing many of the things we've said about science versus art.
I don't think anyone I've ever asked ever said be a filmmaker. Maybe virologists don't want to be filmmakers, but other scientists do. But I would never want to be a filmmaker. From what I see, going into making a film. Man, I don't need that. However, I do want to make short videos on my own about science, but that's not being a filmmaker. Wow.
Yeah.
Alan, what do you have for us?
I have, what did I pick?
The better boarding method.
Yeah.
This is great.
So this is a video. This is a YouTube video using some, discussing some math, but in a really fun way, about how airlines should board. Mm-hmm. And this is a complex problem, but it turns out that there are solutions that would work a whole lot better than the current approach that most airlines use. And then the video explains why the airlines won't use those methods. But it's just a really fun, well done video.
Yeah, I saw this a couple of weeks ago. CGP Grey is a well-known internet video guy and has a lot of.
It's well animated, well narrated.
Yeah,
and
it's
just
good.
Really good.
Yeah, now
I like it.
We could board a lot faster if they used this method. But
I will say Southwest comes pretty close to to one of the more ideal methods.
Yeah.
But they still have kind of a little scam going on too.
Dickson, what do you have for us?
Well, I would like to widen the scope of my interest away from National Geographic photo winners to something that relates to saving the world. There's a new building material that's probably as old as the oldest building material, and that is wood. And I would like to call your attention to a building that was constructed in Minneapolis recently called T3. This building is using the laminated wood method for creating beams that are then fitted together just like you would a timber frame home. And timber frame homes go way back into the Japanese history, which is where they originated, as far as I can tell. And this building has metrics associated with it that'll just blow your mind. And one of them is how much carbon they've sequestered in the building simply by building it out of wood. And the answer is 3,646 tons of carbon are sequestered in that one building. Now, the advantage of a wood building over a cement and steel building is that when you want to repurpose the building, it all comes apart just like Tinker Toys.
You can just take a match to it.
Well, you'd think so, Alan, but try that sometime. And they actually showed you a demonstration.
No,
you can't just put a match up against a tree and set it on fire.
Right.
And that's the same principle. You can char it, but you can't set it on fire. You need some smaller pieces of wood to increase the surface.
You need to light the furniture first.
You do. You will light there. You go. This building is also clad in a thin skin of iron to prevent the invading Huns from Central Europe coming with flaming arrows and tar at the end. That won't.
But also
either.
the termites.
But also the termites. They're not worried about termites. In fact, these are termite-proof buildings.
Oh, right. They did laminate. Yeah,
they did, and the ants won't drill into them, and the driller bees won't drill into them. So. This is the new building material that all the architects are talking about. And imagine an entire city made like this. And so at Fordham, I'm directing a class where we're actually going to do that. And the metric for that city, I will tell you at the end of our exercise.
This is in Minneapolis.
Yeah.
Where we're going to be for ASV this summer.
Well, you could visit the building.
Mm-hmm.
This would be fantastic. And the building codes prevent some states from actually having a lot of these buildings because there are unions that actually relate to the iron and steel workers and cement and which prevents these from happening. But Oregon has just eliminated its entire code and will permit anybody to permit anyone to build a building like this. In Japan, they're designing one 70 stories tall.
Oh, I was just going to ask you if there was a limit. Yeah.
There's none. There's no limit.
Do they have to have that ironclad?
They don't. No, they don't. They do not. They do not. So if you go online and just type out mass timber buildings and go to Google Images, you will be blown away by the number of designs that are out there right now. It's incredible. And guess what you get 20 years later? If you built an entire city out of this stuff, right? And you wait 20 more years after you're finished building your buildings, the forest has grown back. So you've doubled the carbon sequestration.
Where you took the trees from to begin with,
Correct.
you mean?
Right.
Yeah.
That's correct.
Are there any of these in New York?
Not that I know of. Not that I know. I think there are building codes against this and for height.
I see.
but not in certain states. In Minnesota is one of those.
We should have our studio in this building.
We should. And the interiors of this building are incredible. There's so
plenty
beautiful.
Of weather to talk about, too.
That's right. And you can see how old the building is simply by counting the rings. No, that's not right. No, it's quite an amazing turnaround.
Cool. Very good.
Yeah.
Kathy, what do you have?
I picked something that is kind of fun. It's a flow chart for which living things should you study? And it starts out with, do you like waking up early? And if you like waking up early, then maybe you should study birds. And then it goes through a whole series of things. But one of them is, do you love peering at tiny stuff with a hand lens? And if the answer is no, then you go down to, would you rather peer through even bigger instruments at even tinier stuff? And if the answer is yes, then that leads you to microorganisms. But
I
it's
love
really
the bit
fun.
at the bottom.
Oh, yeah.
I don't have to follow your rules. Fungi.
Right. So it's just
That's
really
great.
fun to work through.
Yeah, that's really cool. Nice.
Good.
Very nice. How did you find that?
It was in the ecology and evolution blog that I follow, and at the end of each week they have a whole series of links, and that was one of them. I think.
My pick is an article on a website that I don't usually look at. It's called The Air Current. It is by John Ostrower, who writes about aviation safety. And the article is, the world pulls the andon cord on the 737 MAX. And as you know, there have been 2 terrible crashes of this new airplane recently. And this is an article about what happened and why Boeing made this plane and the economic pressures it was under to make, a new 737. And they put big engines on it, and it pushed the nose up. So they had to make the software that pushed the nose back down. And apparently it went off in these 2 crashes. So it's kind of interesting if you want to know what's going on.
This is a really good article. I followed the technical aviation aspects of this, but I wasn't familiar with the business background of it. And this guy's reporting really puts everything in context.
So, Alan, basically the software seems to have gone off. Is that right?
Well, yeah. So what happened in the Lion Air crash? The software is supposed to trim the nose down to make the plane handle normally, because they've got these huge engines they put under the wings, which cause the nose to pitch up, especially when you're taking off. And so they've got this system, the Maneuvering Characteristics Augmentation System, or MCAS, that trims the nose down when it detects that the angle of attack, that the wings are at too high an angle. And in Lion Air crash, the angle of attack indicators were 20 degrees out. There are 2 different ones, and they disagreed with each other by like 20 degrees, which should have been detected as a fault, but the equipment to detect that and indicate the fault to the aircrew is optional. So, they hadn't bought that option on the package, and so they didn't know that the system feeding into this was not giving good data to the computer.
Wow.
And they also didn't know that the MCAS system existed because Boeing didn't mention it in the manual. They decided that since this wasn't going to be anything the pilots would ever need to really interact with, they just wouldn't mention it. Which is really the crux of the problem, because it's possible to disable this thing, but you have to know that it's connected to the electric trim switch, and you have to disable that. And if this happens on takeoff and you're not expecting it, and suddenly the plane is pushing its nose down and the pilots are fighting it, they turn off the autopilot. That doesn't disable this system.
So, Alan,
it's going to crash the plane into the ground, and you've got a couple of seconds to react to it. And if you don't know what to do because it wasn't mentioned in the manual or the training, then this is going to happen. It's not that the plane is bad. I mean, if this is what happened in Ethiopia as well, it's not that the plane is a bad design or that this is an inherently unsafe system. It's just that the way they rolled it out seems to have been suboptimal, to put it mildly.
Yeah.
So I have a couple questions, because one is that when there's a new airplane that comes out, the simulator has to include all of the possibilities, including the nose up, because there's too much thrust. Sure. And you have to train on that for so many hours before you go into the new airplane. These pilots didn't do that?
This was not
It's not part of the simulator?
It's not drilled in because it's not part of the documentation that Boeing provides for training on the plane, and they didn't think that people needed to be trained on the details of this system.
But I thought the simulator crashes on purpose.
They didn't anticipate this failure mode.
Ha!
And now this failure mode has been discovered.
Yeah.
And this is my understanding of it, is that, and of course, Boeing has its own explanation that, that's not their fault and their plane is fine. And they have a good point. It's a good plane and it's properly designed and it's safe if flown properly. But there's a gap here in the manual and in the training that they promoted. And now, of course, all the major airlines after the Lion Air crash, they went and made sure that all their pilots knew about this. And I don't anticipate that to be a problem for most of them. If it happened again in the Ethiopian crash, then, maybe they weren't up to speed on it or weren't ready to respond to it. But there is certainly the appearance of a problem here.
So I asked my wife going downtown one morning recently, how many takeoffs and landings has this new airplane undergone altogether throughout the world? So you have to know how many planes there are, how many flights per day, all this other stuff. But that number was known. And this, if you looked at the odds, it's almost like 1 in 2 million people get sick from a vaccine. You'd say, well, there's nothing wrong with this airplane because for the 99.999% of the time, it doesn't fail.
The statistics on it are really impressive.
They are.
Global airline travel is the safest means of movement ever devised by humans, and the 737 is one of the safest airliners ever built.
Still.
Including the MAX, including the recent crashes.
Exactly,
This is still way, way ahead of anything that came before.
Right.
But
so if you're Boeing, how do you respond to this?
The reason why that's true, why aviation has such an incredible track record, airlines particularly, is because of this obsessive focus on safety.
Agreed.
That is exactly why you have things like the FAA pulling the chain and saying, okay, we're going to ground all these planes, even though they're very, very unlikely to crash. And you're safer traveling in that than you are driving to the airport. But our approach to safety in aviation is that we don't mess around with this stuff. And if there might be a problem, you don't take off until you figured out what the problem is.
Sure. So China said, we're not buying any more of this airplane. You know what we're going to do? We're going to build our own.
Yeah.
And our own will be safer than this.
I don't know.
Well, that's what they said.
Right.
That's fine. Well, this could be a big problem for Boeing.
Exactly.
But anyway, great pick.
It is a good article.
Sorry.
Yeah, wonderful pick.
I have 2 listener picks. One is from Richard, our DDS listener. He sends an article in The Atlantic, The Real Cost of Knowledge. As many of you know, University of California has decided not to renew its journal subscriptions with, Elsevier, right?
Elsevier.
Yes.
Because they said not enough open access. And I think this is a good decision. I put this on Twitter a couple of weeks ago, and someone said, yeah, but I feel bad for the grad students who can't read the papers. But you have to start somewhere, right? Because if you're going to break this, you have to start. And this is a big university system, right?
Yep.
So I think this is a great idea. The Real Cost of Knowledge. And Islam sends a cool poster from
it's very cool.
PDB 101. So, the protein data bank is where you put your coordinates for structures. And there is a educational portal called PDB 101. They have great images all the time. And this is a lovely poster, 200 icosahedral viruses from PDB, which you can print out, 18 by 24 inches. And it's got all of them in different colors from small to big, including maybe the 8 we talked about today.
Maybe. Might be over there on the left-hand
T=1.
end.
Yeah.
T=1. And they got the different capsid subunit numbers at the bottom, right? The number of protein chains from 60 to 960 to 8,280. It's really beautiful. Lovely. You're going to have to print this out, Dickson, and just wallpaper your house with it.
Why not? At least the bathroom, because I spent a lot of time in the bathroom.
No, TMI.
We did.
TMI.
No,
We don't need
combing
to know.
his hair.
Yeah,
Exactly.
combing your hair.
Combing my hair.
brushing
The
your teeth.
hair. I'm combing the hair, not my hair.
Don't brush your teeth. It causes a bacteremia.
That's what you told me. And then it causes Alzheimer's. I don't
Yeah.
want to go there. I'm sorry. What did you say?
Porphyria gingivitis. All right. TWiV is at Apple Podcasts, microbe.tv/TWiV. You can find it on any podcast player on your phone or tablet.
Or on your Apple TV.
It's also on your Apple TV. Yeah, you can do podcasts there as well.
You can.
I don't know about Roku though, since I don't have one. But and people ask, why aren't we on what's that music thing? Never mind.
Spotify?
Spotify, yes. We're on. TWiV is on Pandora, but I don't know about Spotify.
I think we are on Spotify. I think my students have told me.
Because I tried to put it on, but I had to join to do it, and I said, no, I don't want to join.
Got it.
But, I mean, no. And you just subscribe. We would like it if you subscribed. And that tells us how many people are listening. It's helpful for us. And if you have questions and comments, we really enjoy it. TWiV at microbe.tv. And if you really, really like what we do, even a little, consider supporting us financially. You can give $1 a month. That's nothing, less than a coffee, right? You can't even get a cup of coffee for $1 anymore.
Much less.
But you can get 4 TWiVs a month.
Whoa.
A quarter each. There are other things you can do as well. You can go to microbe.tv/contribute and learn more about that. Dickson Despommier can be found at Racaniello.org. And here's a good one, Fortimopolis.com.
There you go.
Is that it?
Yeah.
And that's where his students build new cities.
Indeed.
Can we live in them?
I hope so. That's what we're doing. That's why we want to.
Thank you, Dickson.
You're welcome.
Brianne Barker's over at Drew University, which is in New Jersey, and on Twitter, she is at BioProf Barker. Thank you, Brianne.
Thank you. It was great to be here. I really like these papers.
Kathy Spindler is at the University of Michigan in Ann Arbor, Michigan. Thank you, Kathy.
Thanks. This is a lot of fun.
Alan Dove's at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.